Okay, so for this video, we're sort of merging chapters 13 and 12 and 13. We're kind of streamlining it a little bit. We're going to talk about the basics. We're going to continue this discussion about transcription translation, go into the details of translation, the genetic code, and then talk a little bit about types of mutations towards the end of this PowerPoint or towards the end of this video, and then also a little bit about different ways that mistakes get repaired in DNA. Okay, so just, in case it's been a little while since you listened to the transcription video, let's just go back and look at transcription translation, kind of where it's happening in a eukaryotic cell. You see transcription there happening within the nucleus. We've taken out, a bunch of the details of the processing of the pre-messenger RNA molecules and things like that. We'll just say, okay, transcription. You have an mRNA molecule, finds its way into the cytoplasm. It's there in the cytoplasm that translation happens. You see there at the bottom the yellow sort of glob. That's a protein being synthesized from the mRNA molecule. So this is basically, what are the end results of both this transcription and translation. We're going to talk a lot more about the detail and a lot of the other molecules that are involved in translation. But first, let's just have a brief discussion about protein structure and how proteins are made, or what they're made up of, rather. It'll be a bit of a review maybe of some of the stuff that you learned when you did the organic molecule section in, some of you in zoology 1110. So proteins are complex molecules or polymers in eukaryotes. Well, actually, first in prokaryotes, the proteins or these polypeptides are made up of 22 different amino acids. In eukaryotes, we usually talk about 20 different amino acids that make up our proteins, most of which we're able to synthesize within our bodies. 9 of them we need to eat, right? Those are what are called the essential amino acids. And then there's a 21st amino acid that is sort of a you'd say a modified amino acid. It's called selenosystine, and you can read about that one in chapter 12 on page 315. It talks a little bit about that. But for our intents and purposes, let's just think of the 20 different amino acids that make up the proteins in eukaryotes. Amino acids have a specific structure. Polypeptides are obviously made up of a chain of amino acids. The amino acids are coded for by the DNA, but they have a general structure, the characteristic of each individual amino acids, which will contribute to the overall configuration or conformation of a polypeptide chain, is determined by this side group. The side group that we designate here as R. Also, you have an amino group and a carboxyl group. The interaction between that carboxyl group of one amino acid and an amino group of another amino acid is what gives you the bond between the two different amino acids. Here's a molecular structure of the 20 different amino acids. As you can see, they're grouped into 4 general sort of groupings, with the exception of the methionine, which doesn't group up with any of them. But you see one group has a nonpolar side group that makes it hydrophobic. Another group has a polar side group. The polarity of that makes it hydrophilic or water-loving, right? And then you can either have a basic or an acidic side group. So those are the 4 characteristics. Obviously, you don't have to know all of the different molecular structures of all of the different amino acids, but just understand that this is what contributes to the eventual three-dimensional shape of an amino acid, the function possibly of enzymes and things like that, and um, just kind of understand these contributions. As I mentioned before, the interaction between the carboxyl group and the amino group of two amino acids is what forms that peptide bond. The peptide bond is that bond between amino acids in the construction of the protein polymer. So the interactions of these side groups, or one amino acid to another, gives rise to secondary structures. Here in the slide it shows you alpha helix secondary structure. Another one is called the beta sheet secondary structure. Secondary structures then interact with each other to form tertiary structures, and then tertiary structures interact with other molecules. Here is an example of hemoglobin to give you quaternary structures. And so if you remember what you learned about enzymes, a lot of the functions of enzymes depend on that three-dimensional structure. And so this three-dimensional structure is important, or rather the interactions between these amino acids is very important for the final three-dimensional structure of the protein, especially for the with regards to the performance of something like an enzyme. And so if you switch an amino acid in that chain, it can have sort of a cascading effect on the three-dimensional structure of the protein. This slide just kind of shows you some of the molecular interactions, or the type of bonds that will form between different parts of the amino acid chain that is influential in determining those three-dimensional structures. Okay, so now that we're done with just a little brief background on amino acids, molecular structure of amino acids, etc., let's now transition to talking about translation. Okay, so we know what amino acids kind of are. We know that the sort of that first step in gene expression is transcription. We talked about that in the last PowerPoint, and so now let's jump right into translation. So translation is sort of that last step where your genetic information finally gets converted into something useful, something real, and tangible. Remember, though, that sometimes the RNA molecule actually serves as something real and tangible, but in most of the cases, it has to pass through, that information has to pass through translation. The copy of that information, again, is this mRNA molecule that we talked about during the last PowerPoint. So transition is a fairly complex process and it requires a lot of different players, a lot of different molecules. This slide just kind of shows you some of the molecules that are necessary for transition to take place. The information on how to produce each of those things in the previous slide ultimately has to come from our DNA, and this slide shows you the production of some of these things, like the RNA nucleic acids that have to combine with the ribosomal proteins to build the ribosomes, and some of the tRNAs and things like that. So this is a nice little schematic just to sort of show you where all these things are coming from. We should mention that ribosomes, are not all the same. We have different types of ribosomes, and in particular, we have distinct ribosomes that we find in prokaryotic cells versus eukaryotic cells. This slide shows you a little bit more detail about transfer RNAs. The important thing to look at is there at the bottom, not necessarily the sort of the shape of it, but given this shape, there's sort of two important ends. One end contains the anticodon. The anticodon is what will bind to the codon in the mRNA molecule, and then the other end there, if you look real close, the 3' end, that is where the amino acid is covalently attached to the tRNA. And so this is the molecule that really is where is sort of the core of translation. This is where the translation is happening. It's happening between the anticodon binding to the codon of the mRNA and the very specific amino acid that is attached to that tRNA with that very specific anticodon. And we'll see this play out here in a second. It is very important that tRNAs are very specific, and here this slide just sort of emphasizes that point. This slide brings all of those different molecules together. You have the tRNAs and this one in fact shows you 3 different binding sites within the ribosomes where the tRNAs will come and bind. When we go and talk about the process here in a second, I think we just show 2 binding sites. This one is a little bit more detailed. We have an exit binding site, we have a peptidal binding site, and then we have an amino acyl binding site. So this is where it's all happening. You have the tRNA with those 3 sites that I just mentioned, and you have the ribosomes, or the 2 different ribosomal subunits, the 50S and the 30S subunit, latched onto the mRNA. And you have the amino acid chain being formed and sort of snaking its way away from this what we call a complex. Okay, so now let's go and we kind of have all of the background done. Let's just go through and in a more sort of simple diagram go through exactly the process of translation. So these are the sort of the simplified icons or drawings that we're going to use as we go through the whole process. On top there you have the messenger RNA showing the different base pairs. The first 3, AUG, this is a codon. Remember, a codon is 3 base pairs. The sort of 3 base pair currency or unit that gets read. The AUG is what we call a start codon. Then below that we have the ribosome that is consists of a large subunit and a small subunit, and then sort of the cartoon of the transfer RNA showing the anticodon there at the bottom and the attached amino acid. Again, this figure just showing the codon. It's important to note that the ribosomes are made up of these 2 different subunits, the large and small, and that they kind of act independently. So they're not both going to necessarily attach to the mRNA molecule or to an initial tRNA molecule at the same time. They do it at different times. So this 3 base pair long sequence called the anticodon is going to associate and bind in a complementary strand, or in a complementary way, like we have seen with transcription and DNA replication, to the codon that is located on the mRNA molecule. And so it's this codon anticodon association, anticodon amino acid, specific amino acid association is really the nuts and bolts of the translation process. So this is the genetic dictionary you use to translate protein, right? Or translate, rather, mRNA codons into protein. And so the codons there, the bases there, are the bases that you'll find in the codons of the mRNA molecule. And so we've talked about AUG, that codon, before. If you look there in white, you see AUG. That is the start codon. That's where the first amino acid begins to start making that polypeptide. And so that's a methionine. That methionine sometimes can remain there as the first amino acid in a protein, but oftentimes it gets kind of excised out. How you use this dictionary is the left column is where you read the first base in the codon, the upper part, the top of the chart is where you read the second base, and then the third base is read within the box, each of those boxes on the right hand side there. So just as an example, say we had a codon GCG, we would look for the G there at the bottom left hand corner. We then look at the top of the chart and find the C. You then see that there's a little box with white dashes around it. That is sort of that GC box. Then within that box, we look over to the right hand side and we see which one of them has A, or in practical terms, you just kind of look at where's the GCA in there, and what we see is the GCA codon codes for the amino acid alanine. Okay, so instantly we see, oh, well, there's a whole bunch of codons that code for alanine. This is true, and this is what we call a redundancy in the code. What this means is that more than one codon can code for a specific amino acid, but no codon will code for or translate into more than a single amino acid. So you should be able to take a stretch of mRNA, be given a stretch of an mRNA molecule, and be able to go through, find the start codon, and then go through in a codon per codon fashion until you get to a stop codon. There in the white upper right hand corner, you see the stop codons, and go through and translate a piece of mRNA. So most of you probably have done that in Zo 1110, and you'll be expected to be able to do that. Obviously, you don't have to memorize the genetic code, right? But you should be able to take it through the DNA, mRNA, tRNA, and protein. This slide just kind of takes you through that process. And here's a little practice slide that you can do it yourself. For simplicity's sake, though, I've already divided it into the codons, and if you look at the first stretch of DNA, the TAC, you will notice that is the mRNA that is going to be transcribed from that is going to be an AUG, and fortunately that's the start codon. So you don't have to necessarily find the start codon on this, but be able to go through and translate stretches of an mRNA molecule for the exam. Okay, so let's just go through the basics here. The first thing that has to happen is you have to prepare the tRNA. And so what the tRNA does is that it first attaches to the specific or aspecific methionine, in this case, amino acid. You then have that sort of tRNA attached to the amino acid, then bind to the small ribosomal unit. This is what we call the initiation complex. That initiation complex then binds to the mRNA. So like I mentioned before, when we were talking about the ribosomes, the smaller ribosomal subunit and the large ribosomal subunit actually act sort of very differently. And it's not until all of these things have been bound together, the tRNA, the methionine, and the mRNA molecule, that the large ribosomal subunit comes in and latches onto it. And then you have everything sort of ready to go. The ribosome will then begin to move along the mRNA molecule in sort of a ticker tape fashion, reading the codons, which are 3 nucleotides, at a time, and then inserting the amino acids so they start at the AUG codon until they hit a stop codon. And here on the slide, you can see it listed the stop codons. And this sort of basic process happens in all sorts of organisms. The DNA code is fairly, for the most part, we'll just say it's universal. But then also I want to point out that this not only is going on in our cells with regards to the nucleus and the cytoplasm, but also in organelles that have their own DNA, like mitochondria and chloroplasts. The next 6 or 7 slides are just sort of snapshots of the main things that are happening. And so I put these in here just to kind of give you a good idea of, okay, in each of these steps, which are really just, part of a continual process, what are the sort of the basic, most important things that I want you to remember? Okay, so here, this just shows the formation of the initiation complex. The initiation complex binds to the mRNA molecule. Notice the start codon there. Here we show the different sites, the binding sites, the catalytic sites. As we mentioned before, this is sort of a simplification of it. But then notice, on this one at least, notice the catalytic site. That's where the peptide bonds are going to form between the amino acids that are being carried in by the tRNA molecule. We are now done with the initiation stage, and we begin the elongation stage. So a second tRNA comes and binds the anticodon of that second tRNA molecule, binds with a codon of the mRNA molecule. And the peptide bond forms. Now you get the ribosome moving to your right, again in sort of this ticker tape jolting motion, opening up now a binding site, the catalytic site, and then releasing the tRNA that now no longer has the amino acid bound to it. The third tRNA now comes in, and we cycle back through what just barely happened. Peptide bond now forms, and we again keep going through until eventually we hit the stop codon. Once we hit the stop codon, there is no amino acid to bind to the tRNA that is going to sit down on the codon on the mRNA, and so then you no longer have an amino acid that there. That ends the elongation process. Here you get to the termination stage, signaled by the stop codon. There's a stop codon. You don't have a tRNA that's going to bind to the stop codon. You actually have a protein called the releasing factor that we'll show you in the next slide. But essentially what happens is you get there, and then everybody, quote unquote, basically goes their own way. You detach, and you now have your completed peptide or amino acid chain. This slide just shows that release factor. Here is another slide just to kind of show you it all from the beginning. So the transcription and the translation, basically going through the main part of what we call the central dogma, taking genetic information and making it into proteins. Here is a link to a nice little video that kind of shows you transcription and translation as sort of a moving, real process. So sort of the final aid in helping you study for this. Okay, for the last section we're going to talk about mutation, the importance of mutation for the evolutionary process, and then also a little bit about how DNA is repaired, how mutations are repaired in the cell. So, mutation and evolution, it's important to remember that mutation is sort of the ultimate source of all genetic variation. You do get recombination after you can get new combination of things through stuff that we've talked about, through crossing over and through meiosis and things like that. And now even you could probably say you get some variation based on how your different genes are expressed through epigenetics. But ultimately, all of that difference has to come from a mutation in your blueprints, in your DNA. The more favorable combinations, or the more favorable combinations of this genetic variation is going to be selected for by natural selection and then passed down to offspring. So in this way, evolution depends ultimately on mutation. And that's why, if you remember at the beginning of the semester, we talked about, okay, what was one thing that whatever the genetic material was that it had to do, it had to be able to change. It had to be able to change, because without the ability to change, you're not going to be able to adapt, and you're not going to be able to evolve. Mutation is a stochastic or largely stochastic process, meaning it's basically random. It's non-adaptive in most cases. Sometimes it obviously can be adaptive, but in the vast majority of the time it's non-adaptive. And we sort of break mutations up into two categories. One category that's called spontaneous mutation, and the other category that's we call induced mutation. Induced mutations are caused by mutagenic or mutation-causing agents. We're familiar with a lot of mutagenic agents, and those are a lot of times things that we try to avoid as best we can. Mutations can also be categorized as germinal mutations or somatic mutations. Here's a short list of some known mutagens. Spontaneous mutations are generally quite infrequent. As you can see here, they do happen a little bit more in bacteria and phage, viruses also, than they do in eukaryotes. If, however, you expose organisms to mutagens, you can increase that mutation frequency dramatically. In some studies of bacteria, we see that you can increase it to the point of where they happen at a greater incidence than 1%. So that's per gene. So that's a pretty good rate. If you remember back, or you can even flip back to our discussion on the genetic code, we mentioned that the genetic code is degenerative, or it's a degenerate code. This means that more than one codon can code for an amino acid. Because of this, we have lots of mutations where you throw in the incorrect base pair, and in fact it has, basically a neutral phenotypic effect. What we call these neutral mutations. Maybe a good exercise would be to go back and look at that the table, the genetic code table, and see which codon position, the first, second, or third, is more likely a change in which of those codons is it more likely, or not codons, change of if the base pair of the codon, first, second, or third, is it more likely to change the amino acid. Because of this, we have mutations that are called synonymous or non-synonymous. Synonymous mutations are these neutral mutations. These are the ones that don't have any effect. So you can change one of those three base pairs and you're still going to get the same amino acid. And then non-synonymous means you're not getting the same amino acid, so a change in the base pair did actually, in this case, result in a change in the amino acid. So hopefully you went back and you saw that it's that third codon position that can change quite a bit, and you still get the same amino acid. And then the second codon position is sort of the other end of this short spectrum, right? And you hopefully saw that when you change the second base pair in the codon, the second codon position, you would get an amino acid change. Okay, so we also classify these sort of mutations or these base substitutions by whether a pyrimidine is replaced with another pyrimidine or purine with another purine. This is what we call a transition base substitution. Or if you swap them out, a pyrimidine with a purine or a purine with a pyrimidine. And these are called transversions. And so what we see, especially when we begin to compare DNA sequences between organisms to do things such as estimate relationships, is that we see that these types of base substitutions don't occur with the same frequency. And what we eventually do with those things, with this knowledge, is we begin to construct models of evolution, and then we base our analysis, what we call the phylogenetic analysis, on these models of evolution. Here are some things that influence the rate of spontaneous mutations. In contrast to the base substitutions, which are the kind of mutation we call a point mutation, you can also get mutations where you insert or delete one or two base pairs, and when you do that, it shifts the frame of reading. So if you think about about codons, here on the left you have just sort of an example of some DNA, mRNA, and the polypeptides, or the amino acid, the amino acids and the polypeptide that will result from each of those codons. If you just throw in a base pair, that's going to shift how all of the codons downstream from where you have inserted that nucleotide, how they're going to be read. And so it totally, sort of changes everything downstream of that. These are what we call frame shift mutations, and as you can imagine, they can have some pretty substantial morphological consequences for genes where that insertion or deletion happens to occur. Lots of organisms have enzymes that will scan DNA and look for damaged DNA and initiate repair processes when they detect those damage points. Just to kind of give you a sampling of the kinds of DNA repair mechanisms that are found in organisms, here we have a slide that shows you the DNA repair mechanisms in E. coli. We have a light-dependent repair. The next slide will show you a little bit about light-dependent repair. You have excision repair, mismatch repair, post-replication repair, error-prone repair systems. And so you have a bunch. The book gives a lot of detail in some of these. At this point, I just want you to be aware of these different mechanisms that are at play in our cells and in cells of living organisms. Just so that you are aware of that that aspect of the biology. Here you have a mutation called a thymine dimer, where the thymine base pairs bind to each other rather than binding to their complementary base pairs, the adenine base pairs. This is induced by UV radiation. You have an enzyme called photolase that will come along, detect that thymine dimer, and repair those dimers. And so exposure to UV light, can cause lots of thymine dimers, and so it's nice to have a mechanism that comes in and repairs those for the E. coli. And for the last slide, these are just some examples of excision type repairs of DNA. Okay, so we kind of went briefly through the mutations and DNA repair, but hopefully now, after this chapter and the last couple of chapters, you're at least have a pretty good foundation for what's going on and understanding of what's going on in transcription, translation, and now mutation and DNA repair. Okay.